Hello dear friends and my dear students. Our today's topic is regulation of gene expression. It is a multi-step process by which a gene is regulated and its product is synthesized. Thus, the gene expression results in the formation of a polypeptide. Gene regulation can be at different level in eukaryotes. One is a formation of primary transcript, two is a processing level, 3 is a transport of mRNA from nucleus to the cytoplasm, and 4 is a translational level. The next is Operon concept. It is a transcription control mechanism of gene regulation. In Escherichia coli, when lactose sugar is provided to the culture medium, the cell induces production of 3 different enzymes necessary for the digestion of lactose. These enzymes are beta-galactosidase, beta-galactoside permease, and third is trans-acetylase. The synthesis of these 3 enzymes are controlled by a long segment of DNA which is known as operon. It consists of an operator site O and 3 structural genes Z, Y and A. The action of these genes is regulated by operator site with the help of a repressor protein. This repressor protein is produced by the action of inhibitor gene which is also known as regulator gene. Next is lac operon. Lac means lactose operon of Escherichia coli is a inducible operon. This induces when lactose is present in the culture medium. It consists of following component. So let's take the detailed component one by one. One is regulator gene. This gene controls the operator gene in cooperation with an inducer present in the cytoplasm. Regulator gene produces a protein which is called repressor protein as we had already studied, and this repressor binds with the operator gene and represses or stops its action, which is known as regulator protein. Then number 2 component is a promoter gene. This gene precedes the operator gene and it is present to adjacent to the operator gene. The promoter gene base sequence determine which strand of DNA act as template. Then third component is a operator gene. It precedes the structural gene and this control the functioning of the structural gene. Then fourth is a structural gene. When lactose is added to the Escherichia coli culture, the structural gene catalyzes or produces mRNA which in turn produces polypeptide on the ribosomes. And last one is the inducer which is not a component of operon. It is already written here. So it is a chemical in the cytoplasm which is also known as allolactose which inactivates the repressor, and when lac operon is switched on, then inducer joins with repressor protein preventing the binding of repressor to the operator gene. So these are all important components of lac operon. Then let's come to the genomics. The term genome means total genetic constitution of an organism. The term genomics is the study of genome through analysis, sequencing and mapping of gene along with the study of their functions. They are classified into 2 types. The number 1 is a structural genomics and number 2 is functional genomics. Structural genomics involve mapping, sequencing and analysis of genome, and functional genomics is related to the study of the functions of all gene sequences and their expression in the organisms. Then the application of genomics. The genomics is used in agriculture to develop transgenic crops having more desirable characters. Genetic markers also developed in genomics that have applications in forensic sciences, and genomics can also lead to introduce new genes in microbes to produce different enzymes, therapeutic proteins and some biofuels also. Then next is Human Genome Project. This is really important project. It was initiated by US Department of Energy and National Institute of Health. The countries which took participate in this were UK, France, Germany, Japan, India and China. Those were the international partner of Human Genome Project. It was formally begun in 1990 and was completed in 2003. It was developed to determine the genomic structure of human being. The main aims of the project, number one, was mapping the entire human genome at level of nucleotide sequences. Number two is to store the information collected from the project in database. Number three is to develop tools and technique for analysis of the data. Number four was the transfer of related technologies to the private sector such as industries. Number five was to take care of the legal, ethical and social issues which may arise from the project. The Human Genome Project was to provide a complete and accurate sequence of the 3 billion DNA base pairs that make up the human genome and to find out the estimated number of human genome. So till now about 33,000 genes have been estimated to be present in the human being. So next important topic is our DNA fingerprinting. This technique was developed to identify a person with the help of DNA restriction analysis, is also known as DNA profiling or a DNA fingerprinting. This technique is used or based upon the identification of nucleotide sequence. About 99.9% of nucleotide sequence are same in all persons. So can you believe it? Almost 99.9% of nucleotides are same. Only some short sequences are different from person to person. On the basis of those short sequences, people are identified with the help of DNA fingerprinting. Steps involved in DNA fingerprinting. Number one is isolation of DNA. So only small amount of tissue like blood, hair, root, skin are required to isolate the DNA, and then isolated DNA has been done the restriction digestion of that DNA. So number two step is a restriction digestion. The isolated DNA is treated with the restriction endonuclease enzyme, and the phenomena is known as RFLP, which is known as restriction fragment length polymorphism. Number three is gel electrophoresis. Then the DNA sample loaded for agarose gel electrophoresis under the electric influence, and the DNA bands are formed on the agarose gel. Fourth step is Southern blotting. The separated DNA fragments are transferred to a nitrocellulose filter paper. Then fifth is selection of DNA probe. A known sequence of DNA, single-stranded DNA is prepared, which is called a DNA probe. The probe is labeled with a radioactive isotope. Number 6 is process is hybridization. The probe DNA is added to the nitrocellulose paper containing the host DNA. The single-stranded DNA probe pairs with the complementary base sequence of the host DNA strand, and the DNA hybrid are formed, and the remaining single-stranded DNA probe fragments were washed off. And then last step is a photography. The nitrocellulose paper is photographed on X-ray film by autoradiography. So these are the steps which are involved in DNA fingerprinting. So you can see number first is a sample, and the isolated or the DNA has been purified from there. Then restriction endonuclease or enzyme which are required to cut the DNA into the small pieces. So in the third picture you can see DNA are fragmented. Then they are gone for electrophoresis, where they are loaded on electrophoresis gel or agarose gel. Then they are transferred to the nitrocellulose membrane or blotting paper, which is Southern blotting is known as, and then they are selected single-stranded DNA probes. They are washed off, and then they are gone for the photography. So this is all process related to the DNA fingerprinting, which is really important. Why they are important, or what are the application of DNA fingerprinting? In forensic science, to solve many problems like rape cases or some complicated murder cases, in those cases the DNA fingerprinting is required. Number 2 is to find out biological father or mother or both in case of any disputed parentages. Number 3 is used in pedigree analysis in cats, dogs, horses and humans. So thank you for watching.